Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the lecture number 15, and from today we will start workshop using ImageJ or Fiji for image processing. So there are 3 consecutive lectures will be on this workshop for ImageJ or Fiji, and we will learn about how to use Fiji or ImageJ for your image processing applications. Okay, now before we begin with the workshop, you are supposed to download this software from this link. Okay, here I have provided the link. You can click that link and download the Fiji or ImageJ. So both are the same software, but they are these are the interchangeable names. Either it is known as Fiji or it is known as ImageJ. Actually, the advanced version is known as Fiji, and the original software was known as ImageJ. Okay, you can download this software from this link, and also I have provided some sample images. So those sample images that we will use for the workshop, and they are saved in this folder. Sample images for ImageJ Fiji workshop, week 8, week 9, week 10. So this is the name of the folder which is there in the lecture material folder. So you can download these sample images from this folder. So let us begin with ImageJ. What is ImageJ? So as I mentioned that you can either call it Fiji or you can call it ImageJ. Both are the same software. Only thing, this ImageJ was the original software. It was developed by Vinay Rasband in 1997 at NIH USA, whereas later on some advanced plugins were introduced in the ImageJ, and the new software is known as Fiji. So if the full form is Fiji is just ImageJ. Okay, and that was developed by Shandling in 2012 for biological image analysis. So once you will download this software and you will open it, it just look like this small panel that you can see here on my screen. So this is the software. Okay, though it looks such a small panel, but it can perform thousands of functions. Even after using it for 10 to 15 years, I can say that I only know 10% of its applications. It has so many vast number of applications that not a single person can know the entire software, okay, or all the features of the software. So it provides so many different options for image analysis, and you have to use the appropriate option that is required for your image analysis. Now this ImageJ or Fiji is an open source software, and that is freely available. Okay, so what do I mean by the open source software? So you can write your own scripts. If you know a little bit of programming in Java, then you can write your own scripts, and you can ask the software to perform the functions that you want. Okay, so generally the other softwares, let's say the Microsoft suite of applications that we use, PowerPoint or Microsoft Excel, Microsoft Word, so those are not open source software. So you have no freedom to put any new feature in the software, whereas this is an open source software. So you can write your own script and you can make this software work the way you want. Okay, and it is written in Java, and it is available for Windows, Mac and Linux. So when you will try to install it from the website, then it will show you different versions for Windows, Mac and Linux. And large number of free plugins are available for this software. So let's suppose you need some special kind of image analysis. You just have to search it on the internet that whether those plugins are available for ImageJ, and most of the time you will find those plugins because someone must have used that kind of analysis before. So people generally put those plugins freely available, okay, on the internet, and it has a good amount of community support. So whatever you write, means if you have to learn this software, you just ask the question to Google search bar and you will find a lot of community support. There are several community specifically made for ImageJ support. So you have to write the question that you have and you will get the answer from the community. Okay, and as I mentioned, it is scriptable. So you can write your own script and you can make the software work in your own way. Okay, and there is another version of this software which is known as Micromanager, that is also open source, but this software is used for the image acquisition. okay, not for the image processing and analysis. So Micromanager is also freely available. You can download it from internet, and that software is generally used for controlling your microscope. So let's say suppose you haven't purchased the microscopes, the software that comes with the microscope from the manufacturer, then you can run your microscope using this Micromanager. Okay, so we won't discuss much about Micromanager. We will mostly focus about the Fiji and ImageJ, and these are the software that one can use for image analysis. Now you can see here, just like the other softwares, this software also has menu bar, toolbar, and status bar. Okay, so this one is the menu bar, which has the options like file, edit, image, process, analyze, plugins, windows, and help. Okay, then this is a toolbar. So here you can see the expanded toolbar. So these are the area selection tool. So if you want to make different kinds of region of interest, you can use any of one of this area selection tool. Then this is a line selection tool, and there are several other tools that we will discuss during the workshop. And this is a status bar. So it shows you the status of your image, or if the image is opening, if image is downloaded, if image is processing. So that will the status bar will show you the status of your image processing. Now, what is the advantageous feature of this software? So you know that there are several different microscope manufacturers are in the field. So mostly you will find microscopes from Leica, Nikon, Olympus, and Zeiss. And all these different manufacturers, they save the microscopic imaging data in their software specific format. Like let's say if you have a Leica microscope and you are using the Leica software for image acquisition, so your imaging data will be saved as .lif file. If you are having a Nikon microscope, and if you save your images using the Nikon software, then your images will be saved as .nd2 files. If you have an Olympus microscope, it will save the images as .oib file. And if you have a Zeiss microscope, it will save your images at CZI file. And on top of these four manufacturers, there are several other manufacturers. Let's say Delta Vision microscope saves the images as .dv file. So any kind of file formats, ImageJ can open these files. Okay, you cannot open these files using your regular Windows image processing softwares. Okay, but you can open all these different file formats saved by different manufacturers using ImageJ. So if you have ImageJ installed on your computer, you do not need this microscope specific, the manufacturer specific softwares to open this imaging data. Okay, if someone sends you .lif file and you do not have that software from Leica, still you can open this .lif file using ImageJ. So here is the ImageJ main window, and you can see the menu bar. As I already discussed, the file menu has the options such as open, save, create image. Then edit menu has the options like editing images, drawing, and operations. Then the image menu has the options like modify and convert images, geometric operations. Process menu has the options of point and arithmetic operations and filters. Analyze menu has the options for statistical measurements, histogram plotting, and all. And plugins. So there are several plugins available already pre-existing in the software that you install, but there are several plugins which are available on the internet. So if you need them, you can just download those plugins and you can run those plugins using ImageJ. Okay, so now we'll start using this software. Okay, and this is a workshop. So how we'll proceed that throughout this class, I will just show you that how to use different options and how to do image processing using this software. But later on, you can download those images from the folder that I mentioned on the second slide, and you can practice this thing on your own computer using this software. Okay, so now I will just process the data, and I have mentioned all the steps on this slides. You will see in the subsequent slide also. Whatever the options, let's say I have to go to the file menu, then I have to go to the open samples. So all these things I have mentioned in blue fonts, all the steps I have mentioned in blue fonts. Okay, and now I will show you how to use them, but later on you need to practice them yourself using your computers. Okay, so let us start. First I will show you how to open an image using ImageJ. Okay, so here is the folder where my sample images are there. Okay, so this is the folder that you will download from the sample images for workshop folder. Okay, and for opening a single file, you have to, just like the other software, you go to file, then open, and then it will ask you to open the find the folder. So let's say your folder is on the desktop. I'll go to this folder, sample images for workshop, and whatever the image I want to open, that I can open. Suppose I want to open this actinium image, I can open it like this. Okay, this is the way. Also, you can drag and drop the file in the ImageJ to open it. And if you want to open a series of files, then you have to go to File, Import, and Image Sequence. So you will go to File, Import, and Image Sequence. So let's say you have a time series and you have saved individual time point as a single TIFF file, .tiff file. Okay, so you have, let's say, 50 .tiff file, and those 50 .tiff files are actually a time series of 50 different time points, and you want to open that time series. So what you can do, you can go to File, Import, and Image Sequence, and you can select all the 50 images of the time points, and then it will open all 50 images as a time series. Okay, so that way you can open the image sequences. So there are three ways of opening any imaging file in ImageJ or Fiji. Either you go to File and Open menu, okay, and then or you drag and drop the file, or if the image sequence file there are multiple files, then you can go to File, Import, and go to Image Sequence. Now for this workshop, you will have two sources of sample images which we will use throughout the class. The ImageJ sample images that comes with the software during installation. Okay, so the moment you download this software and install it on a computer, by default it comes with some sample images, and those sample images are present here, File and Open Samples. So you can go to File menu and Open Samples. Okay, so these are so many sample images they have provided just for for practicing on the ImageJ. So let's suppose if I open this HeLa cells, or maybe this fluorescent cell. Okay, I'm opening this fluorescent cell image. So you can see this is image of a fluorescent cell, and I can practice on this image. If also this software provides some sample images for the gel pictures. Let's say gel sample image, open sample, and I'm selecting this gel. So you can see this is a gel picture of agarose gel electrophoresis, and sometimes people even use this software to quantify the fluorescence intensity of these different bands that you can see in agarose gel. So suppose this is agarose gel, and I took this line option and I'm drawing this line, and let's say I want to quantify the intensity of these bands. I can just make the line, and then I can go to the option Plot Profile under the Analyze menu. So under the Analyze menu, you can see there is a Plot Profile option. So if you plot this profile, then you can see the intensity values. Okay, and here where the band is there, it is showing the reduced intensity, because you need to convert this image into black and white. So now the bands are black and the background is white. Instead, you need to invert the image, and for inverting the image, there is an option here in the edit option. You just invert the image. Okay, and then again we can make this line and plot profile. So now you can see. So basically you can measure the fluorescence intensity of different bands quantitatively. Okay, and I have put it on the live mode, so whatever you can actually move the cursor like this, and you can see in the live mode how the fluorescence intensity changes. Okay, so this is also a side application. We are not going to discuss more about this, how to quantify the bands from the gel, but I just showed you that this software can be used for this purpose as well. But our interest is to discuss about the how to process the fluorescence images. So the thing that I want to convey is there are some sample images already loaded on the software. Okay, so you can use these sample images for various kind of practicing and analysis. And the other sample images that I have already shown you, that you have to download those images from the folder sample images for image J and Fiji workshop folder that is given in the lecture material folder. Okay. So now first what we will do, we will go to file, open samples, and we will open this HelaCells.tif file. File, open samples, and HelaCells.tif. This is HelaCells, that is 48-bit RGB image. I will open this image. Okay. Now this is a 3-color image. You can see red, green and blue 3-color. And if you want to see some image information, it is mentioned that press I. So if you press I, then you can see that some image information will come. This is a 16-bit per channel composite color image of HelaCells with red lysosomes, green mitochondria and blue nucleus. Image courtesy means this image was provided by Tony Collins. Okay, and the scale of 19.42 pixels per micrometer is not accurate. So some information about this image is presented in this document. So just by pressing I, here it is mentioned, press I for information. So if you press I, you can see this information about the image. Okay. Now what is the first exercise? What we will do with this image? So this is known as a composite image, composite color image, because it is actually the merged image of 3 colors. Okay. And here on the top, you can see some information about the image. So it is showing 1 by 3. So these are the channels. Okay. So this image has 3 channels, red, green and blue. And now you are on the red channel. So how to select the channel? So here you can see there is a scroll bar with the letter C. So using this scroll bar, you can change the channel. Okay. So you will not see, because now you are seeing all the 3 channels here, but you want to process any one channel, you can just change the scroll bar. So what do I mean by process? So let's say here somehow I'm not seeing the green signal clearly and I want to enhance the brightness of the green signal. So what I can do, there is a tool known as brightness and contrast adjustment tool. So if you go to image, adjust and brightness and contrast. Okay, image, adjust and brightness and contrast. So it will open up this window. And let's say if you want to change the intensity of red color. So now I have selected the channel first channel, that is red channel. So here you can see the graph in red color, and I can change the intensity of that red color. Okay, let's say I want to modulate the intensity of green color. So I will go to channel 2. Now you can see this in green color and also this graph in green color. So with this I can increase or decrease the intensity of the green color. Okay. And now I want to change the color of intensity of the fluorescence intensity of the blue color. So I put the scroll bar on the 3rd channel. So now you can see this in blue color. Also this graph is in blue color. And if you move this thing, it will change the contrast of blue color. Okay. Now see what exactly we are changing here that you see. This is a 16-bit image. Okay. So it is captured by the 16-bit camera. But as I mentioned that your computer screen cannot display more than 8 bits. Okay, so computer screen cannot display more than 256 gray labels. Okay, although your image was captured by 16-bit. So it has the information of 16-bit, but you cannot see those many gray labels on your computer screen. And also this is the size of the image. So it is 34.60 by 26.36 micrometer, and if you want to see the size in pixel, it is 672 by 512 pixels. So in X it is 672 pixels. So if you just zoom it, and you can see the pixels, right? So see, these are each of the pixels. This is 1 pixel, this is 2nd pixel, this is 3rd pixel of the camera chip. Okay, so there are total 672 pixel in this direction and 512 pixel in this direction. Okay, and the size of the image is 2 MB. And one important thing that I want to mention here, so let's say now I am on the red channel, and if I put all these things like this, so it's a 8-bit image, so it's a 16-bit image, okay, and here you can see only 4095 gray levels are mentioned, are shown, okay. So by doing this, I am just changing the display of the gray levels, okay. I am not actually changing the fluorescence intensity. I am just asking the software to display only the pixels which have the intensity, okay. So that way I am not manipulating the data. So let's say I want to measure the fluorescence intensity of, let's say, this structure, okay. Let's say this structure, I want to measure the fluorescence intensity, and just I'm changing the display to make it brighter or make it dull, okay. The fluorescence intensity will not change, okay. And how to measure the fluorescence intensity? You just, let's say you want to see the fluorescence intensity of this pixel, okay. So I'm putting the cursor here, and then you can see the fluorescence intensity here, okay. See, so this is the value. The fluorescence intensity is 1042 gray levels for this pixel, and for this pixel it is 727 pixel, okay. So see, now the fluorescence intensity is 1042, right? Now let's say if I reduce the brightness of this, and then I see the value will remain the same, 1042, okay. That you can see it here. When I am putting this cursor here, you can see the value is shown here in image J, okay. 1042, that is the value for the fluorescence intensity of this pixel. So I am just asking the software to display the pixels, okay. So I am not actually changing the intensity value of this pixel, and that's why this kind of manipulations are acceptable, because we are not changing the original intensity of the pixels. Okay, now our first exercise is you have to separate the channels to the individual colors. So this is a multicolor RGB image, composite image, okay. So you have to isolate the channels. So how you can isolate the channels? Go to image, then color, and split channels. So here you can see the option image, color, and split channels. So if you click this option, you see all these 3 channels are splitted now. This is green channel, this is red channel, and this is blue channel, okay. And now see, from the bottom, that channel bar is gone, because now the channels are separated. Earlier all the channels were merged, so you are seeing that channel scroll bar to change the channel, okay. But now the channels are separated, so that scroll bar has gone, okay. So now what is the next activity? What we'll do with this image? Now convert the images into 8-bit pictures, okay. So now you can see all these 3 channels are 16-bit, okay, because they were captured by a 16-bit camera. But you just want to convert it into 8-bit, because you are not getting any additional information for the visualization, okay, on your computer screen. So for that, and to reduce the size of the image, I just want to change it to 8-bit. So how to do that? So here is the option, image type 8-bit. So I will select this green channel first. I'll go to image type, and now it is 16-bit selected, but I can go for 8-bit. So now it converted into 8-bit, and you can visually cannot see any difference, because as such you are seeing only 8 bits only because of your computer screen, okay. So when you convert any 16-bit image into 8-bit image, you cannot see any difference. And now I will save this image. File, save as, I will save it as a TIFF, okay. Now see, for any microscopic images, generally it is recommended to save this microscopic imaging data in the TIFF format, because TIFF format can precisely maintain the quantitative information available in your image, okay. If you save it into PNG or BMP file or any other file format, JPG file, okay, it will lose some information. So always save your microscopic imaging data into TIFF file. Only save it as a JPEG or PNG when you are making some final images for your presentations. because you will make your presentation, let's say using PowerPoint, and the PNG format is the most suitable format for PowerPoint. So when you want to put this image on your PowerPoint, then only you should save it on as a PNG file. Otherwise, you should save your microscopic imaging data as TIFF file, so that all the information, quantitative information, is maintained. Okay, so I will save this 8-bit image as .tiff file, and in this folder, processed images Gunjan. So I have made this folder, and here I will save this C2 HeLa cell as 8-bit image. Okay, I save it. Now I will go to red channel. This is again 16-bit. I will convert this 16-bit into 8-bit, and I will save it. File, save as TIFF, and C1 HeLa cells 8-bit. Here C1, C2 represents a channel. This is channel 1, this is channel 2, and this is channel 3. Now channel 3 is also 16-bit. I will convert it into 8-bit, and I will save it. Save as TIFF, C3 HeLa cells, and 8-bit. So I saved all these 3 individual channels as 8-bit image. Okay. Now the next exercise is, color images are generated from the array of pixel values based on the lookup table. Try changing the color of your 3 HeLa monochromatic images. Okay, so as I mentioned that generally for microscopic imaging, the monochromatic cameras are used. Okay, so for microscope, this is not a green image, or for the computer, this is not a green image. Okay, this is just a grayscale image, but artificially software has given green color to it. Okay, so this color basically I can change. So there is this option LUT, lookup table. Okay, you can just select this, and let's say I want to see this image in yellow color. I can select this yellow, so you can turn this into yellow color. If I want to see this into magenta color, I can convert it into magenta. If I want to see it in blue color, I can convert it into blue. Okay, if I want to see it in again to green color, I can change. So for all these channels, suppose this red color, I don't like red color and I want to put magenta color, I can go to this LUT and I can change the color. I don't like this blue color, I want to convert it into cyan, I can convert it into cyan color. Okay, so in microscopic images, the colors that you are seeing, those are known as the pseudo colors, because those colors are generally given by the software. Okay, for camera or your computer, this is a grayscale image. Okay, but using this software, you are giving different color to visualize contrasting information. Okay, okay, so you can change the colors using this LUT option. Now what is next? Histograms are used to depict image statistics, frequency distribution in an easily interpreted visual format. Okay, so we'll open the histogram for this image. You can go to Analyze menu and the histogram option is there. So let us select this image. Then I will go to Analyze menu and I will select this histogram option. So in histogram option, you can see this is an 8-bit image now. So it has 0 to 255 gray levels. Okay, and you are seeing those gray levels, and you can see where your most of the pixels are. Okay, and so if you need this data, you can just go to this list. So it will give you the pixel value and the count. So there are 6154 pixels in this image with value of 0. Okay, gray level of 0, and these are the 255 gray level. So there are 722 pixels. Okay, 722 pixels which are having completely bright. Okay, now you can see that there are some small points here towards the end of this graph, and I cannot see them clearly in this kind of representation. So what I can do is I can convert this graph into log scale, so that even these small points can be visible. So see, if I convert it into log scale, then this is the log scale distribution, and those points which are towards the end, I can also see them here. Okay, now what is one incorrect thing with this image. Okay, as you can see, even at intensity value 255, that is the highest gray level, that is a completely bright pixel, you are getting 722 pixels with this value, right? So this image is considered as a saturated image. 722 pixels in this image are saturated. Ideally, you should capture the image in such a way that the entire information is covered within 0 to 255. But here the 722 pixels may have the intensity values even more than 255, but it is because the camera pixel is saturated, and that's why it could not be recorded, or it cannot give you proper quantitative information. So if someone asks me to quantify the fluorescence intensity of the structures, ideally I should not use this image because 722 pixels are saturated here. Okay, let us see that if I make the histogram for this image. Okay, so I selected this, go to analyze and go to histogram. So here you see, and I will convert it to log scale. Here also, and I can make this go to the list. So see here how many pixels are saturated. Still 125 pixels are saturated. Okay, so this is also not a good image for making any quantification, intensity based quantification. Okay, let us see this DAPI image, analyze and histogram. Okay, and you can convert it into log scale. I think here there are no pixel with 255 value. Go to the list and you see just one pixel with 255 gray level. Okay, so this is not a saturated image. Okay, and this can be, ideally it would have been better if there is 0 pixel with 255 gray level, but it's just one pixel, so we can still use it for quantitative application. Okay, so whatever the intensity differences are there, all of them are captured, not a single pixel is oversaturated. Okay, so from this kind of histogram plots, it helps you in making sure that your images don't have any pixels which are oversaturated. Okay, and you can by converting it into log scale, you can see the distribution of this data in a better way, and you have to make sure that none of the pixels are saturated in this image. Okay, now let us see what is the next step. We discuss about these histograms. I also discuss about converting the histograms into log scales for better visualization of the data. Then also we discuss about this brightness and contrast adjustment tool. So this is BNC, that is brightness and contrast adjustment tool. So you can go to image, adjust brightness and contrast. Okay, so this window will appear, and here you can see it is showing 0 to 255 pixels because it is an 8-bit image, and your data is mostly on the left side. Okay, but now you are seeing all the gray levels, but I can ask the software to show me only the gray level. So then if I set this to here, so only the pixels which are having some kind of a fluorescence intensity are displayed. Okay, but by doing so, I am not actually changing the fluorescence intensity, I am just changing the display. So if you see the pixel value, it will not change. So this kind of modifications are generally acceptable. Okay, and what next? Okay, so now we will do one exercise, and let me close all the things first before we move on to this exercise. Just the image should be open, and I think, yeah, there is no other thing open here. Okay, so this is an exercise in which I will show you how to process a 4D image. 4D image means the image which will have X, Y, Z and multi-channels using ImageJ and Fiji. And the ultimate idea of this exercise is to generate the montage that we generally use for the presentations or publication purpose. So let me explain you what is the purpose of this exercise. So I will give you, means we will open one image, that is a 4D image, okay, from the sample images for workshop folder, and then our eventual goal is to make the montage. So what is montage? So you see, generally in the publication or in your presentations, people show 4 or 5 color imaging data like this, okay, with the merged image, okay, and also you have to put the scale bar here, okay. So from the raw 4D image, we will generate this kind of a montage using this exercise, and we'll also put a scale bar, okay. So let us do that. So from the sample images folder here, you open this 4D image 01.db file, 4D image 01.db. Now this image has been captured using a Delta Vision microscope, so you can see the file format is .db. Okay, no other software can open it except you have the purchase version from Delta Vision, but using ImageJ you can open this file. So I'll go to File, Open, and from the sample images folder for workshop, this is the file 4D image 01. So I will click on that file, and the software will ask me that how this file is organized. Okay, so it's a 4D image, so it has information for Z-stack as well as multiple channels. So I will open it as a hyperstack file, and stack order is X, Y, channel, Z, and time. There is no time information, but that's why I have kept it last. But you have to open it in X, Y, C, Z, T. And I will. So this is the default option it is showing, and I'm just pressing OK. So it will open this file. Okay. So this is that 4D image 01.db file. Now you can see a couple of information here. C is 1/4. That means there are 4 channels in this file, but we are currently at the first channel, because you see this scroll bar of channel. This is on the first channel. I can move the scroll bar to second channel, third channel, and fourth channel. So this scroll bar has only 4 positions. Okay, and you can select which channel you want to see. I will also tell you that why you are not seeing anything and how to make something visible here. Okay. Then the second information that is giving is Z stacks. It has currently we are on the first out of 45 Z stacks. Okay. So here you can see another scroll bar which with the letter Z. So there are 45 Z stacks, and it shows you the number at what Z stack you are. So now we are at the 22 number Z stack out of 45. Okay. So this is how the 4D imaging data looks like. So here you have scroll bar to see multiple channels. Okay, so see, now you are at the center of the Z stack. Okay, and that's why you can see the structures. Earlier you were at the one end of the Z stack, so one end of the cell. So that's why you are not seeing anything. But when I put the Z to center, then now you can see the Dapistine nuclei here and some other fluorescence proteins in the other channels. Okay. So this represents the channel, this represents the number of Z stacks, then this represents the size of the image. It's 66.15 micron, and if you see in pixel, it is 1024 by 1024 pixel. So the camera chip is of 1024 by 1024 pixel. Actually, the image acquisition has been done using 1024 by 1024 pixels. The camera chip may be of this size or maybe bigger than that, but we have captured the data using only 1024 by 1024 pixels. And this is captured by the 16-bit camera, and the size of this file is 360 MB. Okay. Now, first what we'll do, adjust the brightness and contrast for all 4 channels. Okay. So first thing is make the fluorescent signal visible in all the channels. So what I'll do, I'll go to Image, Adjust, Brightness and Contrast tool. Image, Adjust, Brightness and Contrast. And now I'll put my scale bar, not scale bar, I'll put my scroll bar of the channel on the first channel, and I will ask the so as it is a 16-bit image, it is showing multiple gray levels, but my data is only in the initial few gray levels, right? So I will ask the software to display only those pixels. So I want to see here the bright spot in this images shows the transcription site, and the faint dots represents the number of mRNA molecules, and that is what I want to see here. So I adjust the brightness and contrast in such a way so that I can see those bright dots also and some faint dot also. Okay, the single molecules of mRNA also I can see. Then I will go to the second channel, and here I want to see the transcription site, and you can see it is showing me all so many 36,130 gray levels, but my data is only in the initial few gray levels. So I will adjust the brightness and contrast accordingly, so that I just want to see the transcription site, as you can see by this 2 dots per cell. Then I will move on to the third channel, and here I want to see one fluorescently labeled protein. So I will adjust it like this so that I can see that nuclear fluorescence. And then I'll move to the fourth channel, that is DAPI channel, where you can see the nuclei, and there also I will push it to this level so that I can see the nuclei well. Okay, so I just adjusted this brightness and contrast for all 4 channels. Okay, now the next step is give colors to all 4 channels using lookup tables. Okay, so for camera, monochromatic camera has captured this image. So for camera, all these images are in the grayscale. But now I want to give colors. So let's say for first channel, from lookup table, I will give the magenta color. Then for the second channel, from the lookup table, I will give, let's say, red color. For the third channel, from the lookup table, I will give, let's say, green color, because this is green fluorescent protein. And in the fourth channel, using lookup table, I will give the color yellow to the nuclei. Although it is stained by DAPI, so generally the color of the DAPI is blue, but just to show better contrast with my other channels, I am giving yellow color to my DAPI stained nuclei. Okay, so my DAPI stained nuclei are in yellow color. Then this is a green fluorescent protein. Then this is another protein which labeled the transcription site, and the single molecules of mRNA are in magenta color. Okay. Now the next step is split channels. Okay, I want to split these channels. Okay, so what I can do, go to image, color, and split channels. Okay, so from one file, it will make 4 files. So this is the individual file for DAPI. This is the individual ZStack file for GFP. This is individual ZStack file for channel 2, and this individual ZStack file for single molecules of mRNA. Okay. The next step is combine all the Z sections in one 2D image. So you want to make the maximum intensity projection image. Okay. So now you can see, let's say here, this is individual file for mRNA, single molecules of mRNA, right? But it has 45 Z sections. You can see all these different Z sections are there. And I want to see all the molecules in one 2D image. Okay, so I want to make the maximum intensity projection image of this 45 Z sections. So how I can make that? I will select this image, and then I'll go to image, stack, and Z project. Image, stack, and Z project. Okay, so if upon opening this Z projection, it will ask me that how many slices you want to merge. Okay, so what I can do, I can actually see that my imaging data, let's say I don't want to merge all the 45 imaging stacks, but let's say my data is from 10 number slice to 40 number slice, then I can give those numbers and it will only merge those many slices. Okay, and there are different types of projections, like average intensity projection, maximum intensity projection, minimum intensity projection, some slices. Most of the time you will be using maximum intensity or the average intensity projection, right? For now, we will go for maximum intensity projection, and I'll press OK. So this is the 2D image. Now the ZStack information is gone, and this is a 2D image which shows the combined information of all 45 Z sections. and you are not seeing something because of the brightness and contrast adjustment. I will just increase the value, this brightness and contrast, so that you can see the things. Okay, so this is the maximum intensity projection image for channel 1. That's why the file name is max C1. Similar thing I will do for channel 2. Go to image, stacks, Z project, and I will merge 1 to 45 slices for maximum intensity. So this is my maximum intensity projection image. I will just adjust the brightness and contrast again for this MIP image. Then I will go to third channel. I will Z project it, stacks, Z project, 1 to 45 maximum intensity. I adjust the brightness and contrast to make the things visible. And this is my fourth Z channel, Z stack of 45. I will merge all the Z planes. Go by going to stacks, Z project, 1 to 45 Z stacks, maximum intensity. And also I will increase the brightness and contrast a bit. Okay. So see, now these are my 4 images. One, this is maximum intensity projection of channel 1, maximum intensity projection of channel 4, maximum, the Z stack image I will minimize. This is maximum intensity projection of channel 3, and this is maximum intensity projection of channel 2. So for all 4 channels, I generated the MIP images. Now the next step is crop an area of 256 by 256 pixel. Okay, on all 4 channels, use ROI manager to save the position of the region of interest, and use the same region of interest on all 4 channels. Okay, so now for the representation purpose, we do not want to show the entire field. Okay, and instead I just want to show some 4-5 cells, okay, in the image. So what I'll do, I'll crop this image into 256 by 256 pixels. So let me take this square tool. This is the ROI, region of interest generation tool. So I'm using this square tool because I want to make the square ROI. I can also make the round or elliptical ROI, or this is a freehand selection tool. So different ROIs I can make. Okay, so now I'll go for square ROI, and I want to make it 256 by 256 pixel. So while I will select the ROI here, you will see the pixel value here in the status bar. Okay, keep watching this status bar here. Okay, so see, now it is 303 by 271 pixel. Okay, so instead of, let's say, 256 by 256, I am making the image for 512 by 512 pixels. So I will select 512 by 512 pixels, or maybe I think let me go for 400 by 400, otherwise it will be too large. So I want to go for 400 by 400 pixel. So you can see now in the status bar, you can see now 400 by 400 pixels I have selected. Okay, with width of 25.84 micrometer and height of 25 by 84 micrometer. So I selected 400 by 400 pixel area in this image. Okay, now the idea is I want to crop this area, and I want to crop the same area for all other 3 channels. Okay, because the same area cropping is required. It should not be that if I go for manually making the ROI of 400 by 400 pixel here, I will end up making the ROI, but it is not necessary that the same cells will be selected here. Okay, so what I will do, I need to save this ROI. So for that, you have to open the ROI manager. So this software also has this option of search bar. So sometimes if you do not know that where this ROI manager is there, whether in the image menu or the process menu or the analyze menu, you just write ROI manager here. So wherever it is, it will open. Okay. ROI manager. Somehow it is not showing me now, but let me go to ROI manager. you can see I can find out the ROI manager by writing ROI here. This is the ROI manager tool. It is in the analyze tools and ROI manager menu. So I can write here and use this tool like this, or you can go to analyze and there is a tools menu, and here you can find that ROI manager. Okay, you can open it from here also. But most of the time, to find some tools, I just use this search bar option because that is easier. You do not need to remember that this ROI manager is in analyze menu. You have to go to tools and then you will find the ROI manager. Okay, so this is the ROI manager, region of interest manager, and I want to save the positions or the coordinates of this ROI. So what I will do, I will press this button, add. Okay, so this ROI, coordinates of this ROI are saved here, okay, in this ROI manager. So now what I can do, the same area, if I want to select on this channel, so I can select this channel and I can just select this ROI. So the same area will be selected here. The same area I want to select on this channel, so I will select this channel and I will select this ROI. So the same area will be selected here, and similarly I will do it on the red channel. So here I selected this channel and I'll select this ROI. So now the same area has been selected in all 4 channels, and the next step is to crop them channel by channel. So let's say I will go to 4th channel first, and there is an option of cropping here, image and crop. So this channel 4 is cropped. Now channel 3, image and crop. So this ROI that we made in all 4 channels, the same size, the same position of ROI will be cropped from all the 4 channels. Image and crop. Okay, so I cropped this ROIs of 400 by 400 pixel, the same area, same size from all the 4 channels. Okay. Now the next step is merge channels. Okay, we want to merge these 4 channels, so we will go to image, color and merge channel 2. So go to image, color and merge channel tools. Okay. So now I have to define, there are different colors are mentioned, and I have to define each of these image for different colors. And so let's say first channel, this max C1 I want to see in magenta color. So this is a magenta. So here I will select that max C1 image. Okay. Then max C2 I want to see in red color. So where is red? Red is here. Max C2. Then max C3 in green color. So here is the green. I will select max C3, and max C4 in yellow color. So max C4. And here I will select none, nothing for blue color, no image for gray color, no image for cyan. Okay, so there are 4 colors, and for those 4 colors I selected those respective images, and I will click this option of keep source images. So it will keep this 4 source images and it will generate a new file, that is a 5th number file, as a merged image. Okay. So this is the merged image of all 4 colors. Okay, you can see the nucleus, you can see single molecules of RNA, and again you can see the scroll bar for channel is here. So if I want to increase or decrease the intensity of some channel, let's say here I cannot see the red color signal and the green color signal. So I want to increase the brightness of red and green. So I'll go to channel 2, it is a green color, and using this brightness and contrast adjustment tool, I can slightly push the green color also, so you can start seeing the green color. Okay, then I want to increase the red color as well. So the first channel, and I can increase the red color also, so you can see the red color also. Okay, this is it. So this is your composite image. It has 4 channels. Okay. Now the next step in the exercise is make montage. Okay, so we'll go to image, stack and make montage tool. So image, stack and make montage. Here image, stacks, make montage. Okay, now before you use this tool, you have to make sure that other images are closed, because it will make the montage of all the open images. So now you can see we have kept this maximum intensity projection images of all 4 channels. On top of that, those original Z-Stack images are also open. So I will close those original Z-Stack images. And only these 4 maximum intensity projection images are open and the composite image is open. Okay. Let me close this original Z-Stack, C2, C3, C4. So now only these 4 images are open, okay, which I want to make montage, and the composite image. Okay, so only those images should be open which you want to include in the montage. So again I will go to image, stacks, and make montage. Now here it will ask me that how many columns and rows I want. So I just want 1 row and 5 columns, because there are 5 images, 1, 2, 3, 4, 5. So I want all these 5 images in 1 row. So 1 row and 5 columns. Scale factor 1. I do not want to enlarge them. First slice, last slice 1, and here last slice I will keep 5. Increment 1, and between the image I want to give a small border, so I'll give the border width of 2, and the label slices and font size I'll keep 12. Okay, and I will press OK. So this is the montage. Okay, this is your merge channel, this is your green, magenta, yellow, and somehow I don't know why the red color hasn't appeared here. Okay, but anyway, there is some technical error so that the red color didn't appear here, but this is how you can make the montage. Okay, now the next step is to put a scale bar of 5 micrometer. Okay, we want to put a scale bar. Now for putting the scale bar, there are 2 steps that we have to follow. Okay, so the first step is we have to define the pixel size to the software, because see what the software knows. Software knows that this image has this many pixels. It has 400 by 400 pixels, 400 pixels in X, 400 pixels in Y. It doesn't know that what was the magnification that we use while image acquisition. Okay, what is the pixel size of the camera chip. Okay, so it doesn't know that how many micrometer, how many nanometers of the sample are covered by each of these pixels. Okay, so that we have to define. So how to define that thing? So what we'll do, we will follow these steps. First step, so go to analyze and set scale. Okay, so go to analyze, set scale, and here distance in pixel is 15. We'll keep the distance in pixel 15, and known distance is 1 micrometer. Okay, and the unit of length is micron, and the pixel aspect ratio we have to keep 1. Okay, so that means 15 pixels represents 1 micrometer. Okay, so we are defining this scale to the software that in this image 15 pixels corresponds to 1 micrometer, and I'll say okay. Okay, and then I have to use scale bar tool to put 5 micrometer scale bar on the image. So go to analyze, tools, and scale bar. This is the scale bar option. Analyze, tools, scale bar. If you don't remember that where these options are, what you have to do is just search here, write scale bar. It will show you that this is the scale bar option. Analyze, tools, scale bar. Okay, and you just have to press this run button so that that toolbar will open. Okay, now here what you have to define, that how much width you want in micrometer. So see, it already put the scale bar here, 5 micrometer. So I mentioned 5, that's why it showed put 5 micrometer. If I want to put a scale bar of 10 micrometer, I can write 10. So now the scale bar is of 10 micrometer. Okay, but let me put 5 micrometer. Height, how much height from the bottom I need. Thickness, font size 14. Let's say I want font size of 10. So see, the 5 micrometer is now smaller, right? So I want font size of 14. Then also where I want to put. and which color. Let's say now it is in white. I want to show it in red color. I want to show it in yellow color. So you can change the color also, but I will keep white. And now it is here, but let's say it's a lower right corner. I want to put the scale bar on the lower left corner. So now you can see the scale bar has shifted from lower right to lower left. Okay, but let me put it on lower right corner only. Okay, and horizontal, if you want to put it vertical also, you can put it the scale bar like this. And hide text, suppose you do not want to mention 5 micrometer here, you just want the scale bar, then maybe in the figure legend you will mention that the scale bar corresponds to 5 micrometer. Okay, so you can hide text also. So those kind of changes you can make, and then press OK. So this is how you can put the scale bar in your montage. Okay, and then the next step in our exercise is save montage as .tiff file and .png file. Now this montage that you will generally use for your PowerPoint presentation or your manuscript purpose. So that you can save as a PNG file. So let me save it. File, save as PNG. Okay, and then I can go to my file folder, and I can, let's give some name that this is montage X. Then I can save it. So I saved this montage as a PNG file. I can directly copy that PNG file and put it in my PowerPoint presentations. Okay. So this is how you can make the montage from your raw 4D image. And see, montage is saved in PNG file, right? But this raw files that you generated from maximum intensity projection and cropping the thing, those you have to save in .tiff file. So let's say later on, if you have to make some changes in this montage, you have this raw files saved in TIFF, where you can change all the things whatever you want. Okay, and then you can recreate the montage. So I'm saving this raw file as TIFF so that later on I can make some changes to the montage. And this is a composite file that I will also save as TIFF. Okay. Okay, so that is the end of this exercise. You please open this image and practice it on your computer after the lecture. Now some more functions, the general functions that I want to show you, how to put the manual timestamp in images. So let me just close all these images first. So suppose you have a time-lapse movie, okay, and you want to put the time scale. How you can put it? So open live.tiff. Okay, so that you will find from the sample images folder. So I'll go to File, Open, and this is my sample images folder. And let me find out whether that live.tiff file is here or whether it is here, live.tiff. Is there any time lapse? So here is that live.tiff file, and I will just open it. Open in the sample images for workshop folder. You will find live.tiff file. Okay, so see, this is the time-lapse movie. Okay, you can see this is a time-lapse movie, and there are 122 time points. Okay, we are currently at the, let's say, first time point of 122 time points. Okay, it's a time-lapse movie, and you want to give the timestamp so that when you play the movie, the time, it displays the time, whether these images are taken at every 200 milliseconds or every 2 minutes or every 2 hours. Okay, so how you can do that? Select color with color picker. So if you want to give some color, you can give the color like, let's say, green. Then go to image stack and timestamper. Image stacks and timestamper. Here it is, timestamper. Okay, so it will open this. Then your time interval, your starting time is 0, time interval is 2 seconds. So you will put 2. Localization, you will keep, localization means at what position in this image you need this timestamp. So we'll define the position as 200, 10, y location 10, x location 200, and uncheck 00 format, suffix second. Okay, so time interval starting is 0 and time interval is 2 seconds. Okay, and I'll press OK. So you see here is the timestamp. And now if you go to the first frame, first time lapse, it is a 0 second, and at every frame it will increase by 2 second, 4 second, 6 second, 8 second. Okay, so if you play this movie, this timestamp will also show you the time point that at every let's say at 70 second this thing happened, at 100 seconds this thing happened, that you can visualize for entire 122 timestamps. Time series. Okay, so this is how you can put the timestamp on your time-lapse movies. Now another tool for object counting. Suppose let's say from this image you want to count the cells. Okay, though here there are just 4 cells, but consider that you have an image with 100 cells in one image. You want to do differential counting, let's say the cells with some signal or cells without signal. So this is the object counting tool here. Okay, so you can double click it, so it will open like this. Okay, and here you can see there are 99, means total 100 counters. This is counter number 0, but if you go for this, there are total 99 including 0, so total 100 counters, and you can define each counter for different type of cell. So let's say I have defined this 0 number counter for bright cells and 1 number counter for the faint cells. Okay, so what I will do, using 0 number counter, I will select how many bright cells are there. This is first bright cell, this is second bright cell. So here you can see the counting. Now I will select 1 number counter to count the number of faint cells. So this is 1, 2 and 3. So with 1 number counter there are 3 faint cells. Okay, so I can use so many different counters to count different types of cells, and those counts will be saved. So if I go to 0 number counter, I want to see that how many bright cells are there, it will show 2. How many faint cells are there? There are 3 faint cells. Okay, so this is how you can use this object counting option for your counting application. Then the distance. So for distance measurement also, you need to define the scale. Suppose I want to measure the diameter of this cell. Okay, so how I can see it? So first thing, this for the software knows that this image has this many pixels. Okay, so this image has 273 by 273 pixel, but software doesn't know what is the size of this pixel. Okay, so I can go to analyze and set scale. So first I have to define the scale, that it's 0.25 micrometer per pixel. Point 0.28 micron. It is inverse, so I think this known distance 1. Okay, so known distance in pixel, 1 pixel is equals to 0.28 micron. Okay, so I set this scale, and then I can go to analyze, tool, ROI manager, and what I can do is, I can, using this line tool, I can make multiple ROI. Let's say I want to measure the diameter of this cell, so I can Okay, so I took this line tool and I want to measure this diameter. So I will do it like this, and this is my first ROI, so I will add this position. Then for this cell I want to measure the distance like this. This is my second ROI. For this cell I want to measure distance like this, add, and I want to show all the ROIs, so I will click this option so you can see all the ROIs. And this is my fourth distance that I want to measure. So I made these 4 regions for distance measurement and I added them to ROI manager. Okay, and now I have to select all 4 ROIs and then I have to click this measure button. Okay, so it will show me the length. So the first ROI is 19.15 micron, second ROI is 20 micron, third ROI is 16 micron, fourth ROI is 18 micron, along with some other intensity values and all these things for these 4 ROIs. Okay, so this is how I can measure the distances using ImageJ. You can save this table as an Excel file and you can use it for further analysis. Now I am not saving it, but you have from the file menu you can save these results. Okay, so I think that's it for today, and the take home message for Fiji, the pros is that it's a powerful and extensible with plugins, and it is scriptable and it has a lot of community support available. It is free software. The cons is that menus are not very intuitive. Plugins are not always stable. Okay, so sometimes some plugins may hang up, or menus sometimes, let's say, you may not easily realize that the ROI manager option is under this menu. So you have to use this search bar to look for the ROI manager or any other option that you are looking for. Okay, so menus are not very intuitive. And one take home message is that bad images cannot be fixed by processing. Okay, so if you put the bad image in, you will get the bad results out. So crap in is equals to crap out. Okay, so for any software for image processing, it cannot make your bad image into good image. It can only increase the visualization of your structures, and it can help you in better visualization. Okay, so ideally for better quantification you need the good quality images as well. Okay, so that's it for today. Thank you for attending this lecture. In the next lecture I will show you how to do image segmentation or the object segmentation using ImageJ or Fiji. Thank you.